All right, so today's lecture is on viruses. And viruses are really interesting because they have some of the characteristics of life, but they actually aren't living themselves. They can't reproduce on their own. They don't have metabolism, but they can evolve. They have their own DNA or RNA. They have their nucleic acids, and they have a protein coat that surrounds them. So today we're going to go over how viruses work, the life cycle that viruses have, and also we're going to talk a little bit about HIV and how HIV infects things and why it's unique compared to other viruses. All right, so a virus's structure, it consists of a protein coat on the outside. Sometimes they also have lipids surrounding that protein coat, but it's a protein coat on the outside, and on the inside you have nucleic acids, either DNA or RNA. And basically what the virus is going to do, it's going to inject its DNA or RNA into its host, and it's going to use the host's own replicative machinery. It's going to use transcription and translation. It's going to incorporate its genes into the host, and it's actually going to cause the host itself to make copies of the virus for it. So it basically hacks the bacteria or eukaryotic cells' machinery to do its own bidding. But the thing about viruses is they only infect certain hosts. So for example, a bird flu or a bird flu virus will not affect a human. Or a dog's flu, if your dog's sick, will not affect you. But you may ask, well, we had bird flu and we had swine flu, we had H1N1, and those affected humans. Why was that? So it's really interesting. Based on the way that viruses actually infect their host, there's special proteins on the surface of a virus that have to bind and match with proteins on the surface of a cell to allow it to go in. What can sometimes happen is if a human flu and a bird flu virus kind of attach to the same cell at the same time, even though normally that bird flu would not affect a human cell, it will allow it to come in and bring it in with it. And then it uses the replicated machinery to make it, the bird flu and human flu, and it actually creates a hybrid virus that can infect both birds and humans. The reason that's so scary for us is that birds, well, we've never seen that specific disease before. So we don't know the effect it's going to have on humans. That's why there's always such a panic about it. You know, people start dying. We don't know if it's going to mutate. We don't know what the effect is going to be long term. So it always worries us when something like this, when a new pathogen, specifically a viral pathogen, comes around that we have never seen before. But normally, a virus can only affect its specific host. The same thing's true with different types of cells that you have. So a cold virus can affect your skin cells, right? Or they can't really affect your intestinal cells, but it affects the cells of your respiratory tract. That's because different cells have different proteins that are made from different genes, as we talked about through gene expression, on their surfaces, which allow the virus to only affect certain parts. Or your stomach flu isn't going to affect your nasal passages, okay? It just depends on the proteins on the surface of the virus, as well as the receptor, the proteins on the surface of the virus itself. So here's kind of showing you what I mean. Here's the virus over here and shows you the receptor on the cell. It shows the virus once it binds to those receptors, incorporating its DNA, and then it makes copies of the virus, and out pops a bunch of more viruses. And this cell effectively is dead at this point. Here's showing you virus B with the receptor B on cell B and virus A with the receptor A on cell A and how they fit together. It has to be specific. Virus B will not affect cell A, and virus A will not affect cell B. Okay, so now we're going to get into viral reproduction. Viruses cannot reproduce without a host, which again is one of the reasons why they're not technically living, aside from lots of other things like no metabolism and things I mentioned before. So there's actually two life cycles that a virus can go through. There's the lytic cycle, which ends up in the death of the host. So this is the virus actually replicating itself, making copies of the virus, and then it explodes out of the cell and goes and infects, hundreds of other cells. So that's kind of the virus doing its job, or it's doing its job to infect your cells. So that's called the lytic cycle. The way I can remember it is like lysis or hydrolysis is breaking open or breaking something. The cell is breaking when this happens and bursting, and out comes all these virus particles that can go and infect other cells. See ya. The lysogenic cycle, on the other hand, is when the virus injects its DNA or RNA into the host. And then those genes get turned back into the host's DNA. It actually goes into the genome, the chromosomes or the circular DNA in the case of a prokaryote, and sits in the host's DNA, and it lays dormant. And it just sits there, and like if the cell replicates, it copies that gene. So the viral DNA stays in the genome of the cell itself until finally, when conditions are just right, the virus will then turn into the lytic cycle again, make copies of itself all at the same time in thousands to millions of cells at once. And then it will, cause like a larger infection. When it is just viral DNA injected that's laying dormant into the genome of the organism itself, it's called a provirus or a prophage. But essentially all it is the viral genes that are just sitting there waiting to be copied until the conditions are just right. That's why you may have heard that if you have a virus, you have it for the rest of your life. And in some cases, that is definitely true, like for plantar warts, for example, or the herpes simplex viruses, like cold sores on your face. They can go away, and then if the conditions are right, they come back. That's because our body fights off the ones that are actively infecting, but there's always going to be some that sit there with the prophages just waiting to express. So you never really truly get rid of a virus. So this is kind of showing you the life cycle between the two. You can see here's a phage injecting now this is again a bacteriophage, so this is showing in prokaryotes, but the mechanism is similar in eukaryotes. It's just the virus looks a little bit different. It's more circular usually. But the prophage injects its DNA into the bacterial cell. Then the bacterial chromosome incorporates it in the lysogenic cycle, and the bacteria can replicate over and over, and it includes this phage DNA in there until finally the conditions are just right, and then it actually goes into the lytic cycle, and it makes copies of itself, and then goes and infects other cells. What's really interesting about this process, so that's the basics basically of how a virus infects human beings, is that when a bacteriophage virus makes copies of itself, the new copies pick up a little DNA of the host cell. And it actually can take it and put it into another cell. So it actually cuts out genes from the host and can infect another cell with it. What we've actually done is we've hacked that process. We create phages that are not dangerous. They don't have dangerous DNA. And what they'll do is we'll make them pick up genes from one host or another, so we can then incorporate it somewhere else. So they actually insert one bacteria's genes into another bacteria. That process is called transduction. This can contribute to bacterial resistance if it picks up bacterial resistance genes from one bacteria to another, specifically to antibiotics. And it can also increase genetic variation within the bacteria too, as well, through this transduction process. So this is showing you bacterial transduction. So here's the bacteriophage injecting its DNA, and you can see this defective phage doesn't pick up the genes of the phage itself that make it infectious. It picks up genes from its host. And now we take that, and that goes and tries to infect another cell, and in fact injects those genes from the original host into a new bacteria, and now this transduced bacteria has the genes from the original one. So this increases genetic variation, and as I said, we can hack this and actually use this in the lab to try to transfer genes from bacteria to bacteria. Here's showing you an actual AP Bio style question. So based on what we just talked about, I want you to try to answer this. I'm going to give you like a minute or so to do it.
What's the answer?
You'll find out what the answer is when that you're going to answer it, and it's going to come up. You know what I'm saying? I can cut this out. So you want to be in this thing?
Yeah, I just want to wave. It's always been my dream to be in an Ed puzzle.
I'm glad that it's been your dream. You have to be right next to me. That you're in it. You can't really see because I don't have my. It shows it afterwards. Yeah.
Okay, just put a clip of this in.
Sure.
It's not too awkward.
All right.
Hi guys.
Put a blooper in at the end.
Hi. Okay.
All right, so the figure above shows several steps in the process of bacteriophage transduction. In bacteria, which of the following explains how genetic variation in a population of bacteria results from this process? We're going to go through each answer. I'm going to explain why each one's wrong and then why the correct one's right. So bacterial proteins transferred from the donor bacterium by the phage to the recipient bacterium recombine with genes to the recipient's chromosome. This one is not correct because it's not the proteins that are being sent, it's the genes, right? We're sending a gene because it's the phage DNA that we care about, so A can't be correct. B, the recipient bacterium incorporates transduced genetic material coding for phage proteins into its chromosome and synthesizes the corresponding proteins. That is true, right? But that's not the reason that the new bacteria gets the new genetic information. It's because it picks up a protein or a gene, excuse me, from the donor bacterium, and then the new transduced phage will inject it in. So B can't be correct. Let's check out C. The phage infection of the recipient bacterium and the introduction of DNA carried by the phage caused an increase in random point mutations. It's not the point mutations in this case that cause genetic variation. Yes, point mutations cause genetic variation, but remember, what is the point of what's going on here that causes the genetic variation? It is D. The DNA from the recipient bacterial chromosome undergoes recombination with DNA introduced by the phage from the donor bacterium, leading to a change in the recipient's DNA. And you can see here that S negative gene and the S positive gene flip. They're just different names. This will become a little bit more obvious when we get into Mendel, but they're just names for alleles, which are versions of genes. So this would be like a functional and a non-functional copy, I guess, and it would switch them back and forth. Okay, so retroviruses are a special class of viruses, and they are very interesting because on the inside of them, instead of having DNA as their nucleic acid, they have RNA. And I kind of mentioned that earlier, that RNA can also be found in viruses as well. But what's so wild about what retroviruses do is that when they inject their RNA into the cell, there's a special enzyme associated with it called reverse transcriptase. Now, I want you to think about what that probably does. So reverse transcriptase takes RNA and turns it back into DNA. It reverses transcription. So what happens is it'll inject its RNA, and then it uses reverse transcriptase to turn it into DNA, which then is incorporated into the genome of the cell that it infects, specifically our cells, because HIV is an example of a retrovirus. So the virus that causes AIDS, it will go into the helper T cells of your immune system, and it will do this process. Now, AIDS is still an epidemic, but it was a huge, serious epidemic back in the in the 80s and early 90s. And the reason that was because retroviruses, when they reverse transcribe the DNA, make tons of mistakes. So it basically mutates the viral genome very quickly. And it changes, because it mutates as the viral genome, it changes the shapes of the proteins on the surface. It doesn't affect the virus's variability or the ability for it to infect, but it does change the shape of the proteins on the outside. So the immune system essentially uses those proteins to identify things that are foreign. So it keeps evading your immune system, and it attacks the immune system itself, so it weakens it, and your immune system can't fight it off. So it's kind of a double-edged sword, or like it kind of whacks it from two different ways. So that's basically how HIV works. And remember, it's a problem, A, because it reverse transcribes the RNA back into DNA. It makes mistakes in doing so, therefore it evolves very quickly, and we can't create a vaccine to fight it off because of that. There's a couple other non-infectious things you probably never heard of. I know this is cut off a little bit, but or at least it's a little, the wording is messed up. But so prions are really interesting. Prions are misfolded infectious proteins that cause other proteins to misfold. You may have heard of prion diseases like mad cow disease. Basically what prions do is they're infectious proteins that can be spread from organism to organism. And specifically in the brain, this happens, a lot of prion diseases. And the protein will then go and cause normal proteins to misfold, which will cause other normal proteins to misfold. This is an example actually of positive feedback, right? We're amplifying the misfolded proteins because as we misfold proteins, more get misfolded, which causes more to get misfolded. And mad cow disease, inevitably, there's no cure for it, because how do you stop a protein from misfolding? And eventually will cause death if you have it. The other thing that, just in case you see it, I'm not going to assess you on it, but a viroid is a circular RNA molecule specifically that cause errors in regulation of plant growth. It's like this infectious little RNA molecule that can be spread. Just in case you see it on the test, I wanted to bring it up. Okay. All right. So that's what you needed to know about viruses and prions and viroids. If you have any questions, let me know.